Hello and welcome. Today I'm delighted to be joined by Professor Danny Altmann from Imperial College, who, together with Professor Rosemary Boyton, are leading our £1.2 million Rosetta Stone research project. This study represents one of the ME Association's most significant research commitments to date. Not only it is our largest investment into ME, CFS and Long Covid research, but it also enables those two conditions to be studied side by side in a way that has never been done before. Professor Danny Altmann is the UK's leading immunologist and a long-standing advocate for people living with ME/CFS. He has been at the forefront of research in post-viral illness, immunity, and long COVID, and his work continues to shape how we understand long-term consequences of infection. Today, we are checking in with him for a quick informal update on how the Rosetta Stone study is progressing 3 months in. So welcome, Danny. For those who are not aware of the Rosetta Stone study, are you able to give us a brief overview of the study and who else is involved?
Yes. So let's start at the beginning. So Rosetta Stone is a kind of metaphor that some people find useful and some people find deeply irritating. But obviously the principle is that, you had this stone that was found and it had 3 partially understood languages on it, in that case some Egyptian hieroglyphics and Demotic and ancient Greek, and by using knowledge from one they could decode insights into the other and therefore decode the whole thing. And I thought it was a useful way of thinking about mysteries in ME/CFS alongside long COVID, because the more kind of nerdy reading I did about long COVID, the more I realized that we were kind of reinventing the wheel. And there were quite specific pathways and clues that long COVID research was throwing up that people had actually talked about for years before in ME/CFS. And I kind of thought if we could knock heads together and knock studies together, we could actually accelerate progress quite a lot. So that's the overview of what it's about. And so what we have is a sort of battery of quite fancy molecular analyses that we plan to do, and I can go into them if people are interested, where really I think almost for the first time, with support of the ME Association, we do a kind of bespoke cohort study side by side of people with long COVID and people with ME/CFS. It's very much led by the insights we got from reading the previous literature and seeing what's looked promising up to now, either in ME/CFS or in long COVID, or shared between the two. So the first thing that comes up is what we call proteomics. And what does proteomics mean? It just means very sensitive detection of proteins in blood, or if you like, biomarkers. So those are the kind of things that can both lead you into understanding pathways and mechanism, but can also give you diagnostic tests. Because one real source of frustration in this whole area is that the reason it's so easy to dismiss people and gaslight people and not refer them for specialist care is because no GP has the right test that they can tick a box for in their clinic and say, here, if you go and get this test, we'll be able to prove what you have, and then you'll be in a treatment pathway. We've never been able to achieve that. So proteomics gets you some of the way down that road to knowing what tests you need to do. And then another kind of hypothesis that's on the table across the conditions is that you have some kind of infectious trigger, perhaps a viral trigger, that leads to autoimmunity, a bit like you might get in, lupus or vasculitis or something like that. And that could be defined by particular antibodies against your own body. And if we had really fancy ways of detecting those, again, we'd find mechanisms, we'd find shared mechanisms, and we'd know what to do about it. So a really kind of heavy piece of work to try and define the autoimmunity in people with long COVID and people with ME/CFS side by side and define those patterns. And that would certainly lead us into new treatment pathways. The next thing that we want to do is that there's some commonalities in stuff that's been published before between gut microbiome changes, bacteria in your gut across the different conditions. So the sort of jargon for that in the trade is dysbiosis, which just means that the kind of bacterial communities in your gut are disturbed. And that sounds kind of trivial, but it's really not, because the bacteria in your gut talk to your immune system in your body all the time and tell it how pro-inflammatory or anti-inflammatory to be, and inform lots of conditions. So again, if we could find the disrupted pathways there, we'd really know what to do about it and could get quite a lot of headway. And then also the other one to talk about is that there have been some quite interesting clues about kind of cellular metabolism and how the cells turn over their machinery in these conditions. And we've got some very fancy kit for looking at that across the conditions. So it's all about getting people in, getting them to come and see us, getting them to give a blood sample and a stool sample, and interrogating these conditions side by side compared to healthy controls and working out what on earth is going on.
Thank you, Danny. So the Rosetta Stone study has now been running for 3 months. How is the study now progressing at this early stage, and what have the first few months involved?
So when you do a big, ambitious, quite costly study like this, what happens is that you draft your grant proposal to the best of your ability, and people peer review it, and they conclude whether they think it's doable or useful or not. And if you're lucky, as in this case, they have the sort of vision to choose to support it. And then you sort of kick it into action. And at the beginning, a lot of kind of quite basic stuff has to happen, where I describe this as the phase where the devil is in the detail, because a lot of underpinning things, if you think about it, need to happen before you can do all those fancy laboratory analyses. Once you start doing those fancy laboratory analyses, data comes in very fast and good stuff happens very fast. But before you get to that stage, if you think about it, you've got to have your chats with HR and write your job descriptions and advertise your posts and recruit your people. And there are timelines for all of that to happen, and how long does the advert have to be out, and how long before you can invite people in for interview and things like that. So we've been through a really exciting phase over the last few months where all of that has been happening. And I've been really impressed by the super people who've applied to us from all over the world. And we've had some really good candidates and done some really rigorous interviews where I don't think I'm jinxed anything, but we've made offers to some really super talented people who I hope are coming on board in the next few months. And that's almost the biggest thing you need to make something like this work, because you can have all the kind of bright ideas in the world, but if you haven't got some people on the ground who are really good at delivering the work, you're just going to be scuppered. So I'm really excited that we've had good people come through the building and good people get interviewed, and we've made offers to them to come join us. So that feels like really a big deal to me. And then you've got to get all your nuts and bolts in place. So to do a thing like this, we've had to tweak our ethical approvals, because although we've done all this kind of stuff before and been doing it for a long time, the second you change things and now want people to send in a jiffy bag with a poo sample in it, or you want them to come and visit you to be tested for their kind of herpes viruses and Epstein-Barr virus and things like that, you need to change the wording on your ethics approval, and the committee need to meet and look at it. So things like that have to happen, and then all of the lab techniques need to be perfected, and so stuff like that. So it'll sound like that's not kind of getting the results through the door, but it is the underpinnings that we need to be in a good place to get the results through the door.
Yeah, I totally understand. You need the right people in the lab to get the results for this study. Like, it's exciting, the first steps. So For this study, will you need to recruit, or will you be using existing cohorts from studies like Wilco, Decode ME, and the UK ME Biobank?
So a bit of both, a bit of all of the above, yeah. So people will appreciate for studies like this, it's really, really important to have the right groups, and so we call them in the proposals, we call them inclusion and exclusion criteria. So that means that when we publish the papers to pass peer review, we want to know that we've got people who really meet the criteria that we claim, or in the age range that we claim, and that we've kind of really compared like with like. So some samples, historical samples, are really important to us. So we've really benefited from the prior investment and the ME Association input to the biobank, and all of the historic work in the biobank. Because if you think about it, we really need that historic build-up, not least because the world has become a confusing place, hasn't it, for studies of ME/CFS since 2020, because there are a lot of people in the world now getting a diagnosis, if they're lucky enough to get a diagnosis of ME/CFS, who have it, almost certainly because they got COVID-19. So this is like the overlap between long COVID and ME/CFS. But if you think about everything I said a minute ago about how we're all about trying to find similarities and differences, if you like, between long COVID and ME/CFS, those kind of people, if anything, are a little bit confusing to us because they're ME/CFS, encompassing long COVID. So sorry, it's a very long-winded answer to explain to you that because of all of that, it's really invaluable to us that there's this historic investment in a tissue bank and a clinical history for kind of the other kind, the long-established kind of ME/CFS, the people who even prior to 2020, perhaps because of an influenza trigger they had or a glandular fever trigger they had, have a very kind of confident diagnosis of ME/CFS that we know isn't due to their COVID-19. And so we can study that kind of phenomenon as well. So we're looking at those groups. We're looking at Wilco, but we very much have our doors open to new people who want to come and see us, who want to be recruited into the study, who either have ME/CFS or have long COVID, or the other group that we're always terribly keen on and find hard to recruit. So tell your friends about it. Is although we talk endlessly about the hundreds of millions of people in the world who have long COVID or ME/CFS, that leaves billions of people in the world who don't. And we really need those as well, the so-called healthy controls to compare with. And healthy controls are the hardest to recruit because they don't perhaps have as much motivation to see this resolved. And we really need people's friends, spouses, partners through the door so we can compare with them. We're very much on the lookout for new people coming into our cohorts, either ME/CFS or long COVID or healthy controls. We're really happy for the ME Association to place that advert on the website. I'll also be spreading it via social media, via various different platforms. And we've also been publicizing the study recently through interviews with Rosemary, Boyton and myself have done for BBC Radio 4 on Inside Health and Inside Science.
Which cohorts will you be monitoring in real time using the Eleros app?
So I think the Eleros app is really valuable to us in the studies. So I think most people know it, but for anybody who don't, the idea is that, again, when it comes to peer review, it can be all too easy to to try and sort of denigrate studies and say these things are subjective symptoms or self-reported symptoms. And my first answer to that is always, well, they have to be self-reported because we just don't have the clinics and the clinical infrastructure for everybody who has these conditions to be reporting them in real time to their GP, because their GP quite often isn't really willing. But what we can do is have really robust, validated, tried and tested applications that really, in a validated way, record people's symptoms in real time and give you a really, really clear picture that passes muster with any medical journal in the world about the burden of these diseases. And Eleros has been developed as a very good example of that. And so when I talk about all our kind of build up at the moment, we're having quite a lot of meetings with the folks at Eleros, because what's that all about? It's about, we want the platform that we develop with them to be really kind of fit for purpose. So if you think about it, what does that mean? It's got to be comprehensive so that this is useful for medical publications and peer review. It's got to not take the people who are using it and drive them crazy by making absurd demands on their time to fill out the same questions over and over again, because we will have lives to lead and want to kind of get on with stuff and not be doing constant questionnaires. And it's got to give a really level playing field for our study so that it's equally suited to people who have long COVID and people who have ME/CFS, and doesn't make any of them feel kind of irritated or excluded by the wrong questions. So that's taken a bit of input to make sure that it's all right. So going all the way back to your first question, like who's in the loop for this, it's anybody who comes forward to our study. And what I mean by that is you could be anywhere in the country signed up to our study and contributing to that really valuable data set. And there's a smaller subset above and beyond that group who are people who are available, I guess usually in the southeast of England, to come and see us at our hospital in West London and give a blood sample. And I'm aware that that's kind of suboptimal, and that not everybody with ME/CFS or long COVID either lives in that part of the country or feels well enough to hop on the tube and come and see us. And I really regret that kind of, to some extent, limits our study, that part of the study, to those people. It's just that if you want to have a study, if you picture it, where you have a whole team of research nurses who could kind of get into trains and buses and cars and drive around the country collecting blood samples, that would be a huge endeavor and a huge expense, and that just hasn't been possible. So we'll do our best.
Thank you for that explanation. So which cohorts will you approach for fecal samples to help the gut microbe investigations, and how will this aspect of the study be conducted?
Yeah, no, I think that's a really important question, because I think we've all read articles in the newspaper or seen things on telly about gut microbiome samples, and it sounds a bit so daunting and scary, but you're like, well, how do they do it? It sounds a bit mucky. And bizarrely for us, you'd be amazed that if you want to get somebody and stick a needle into their arm to collect a blood sample, they don't bat an eyelid. But if they say, and we'd quite like a stool sample as well, they get really terrified, like we're asking something awful of them. So let me explain that it's really not awful. So this is tried and tested over many years, that what we would do to people who signed up is, there are many kind of really nice, hygienic, user-friendly kits out there which describe in really simple, accessible detail how we will send you a discreet brown jiffy bag with a little kind of clinical plastic screw top pot in it and a little kind of poop scoop, and you'll be able to really innocuously and hygienically, in the comfort of your own loo at home, collect the sample for us and mail it back to us in a jiffy bag. So it's really not as scary as it sounds. And what we do then is make DNA libraries from that tiny sample of poop, and use really nice, fancy 21st century methods to comprehensively sequence the bacterial species in that sample, and then do a lot of kind of fancy bioinformatics on the answers, and get really, good stuff out of the analysis on differences between bacterial species in our different groups, right down to how does that impact which biochemicals the different bacterial species might be making, and how that might skew your physiology and your immune response and your inflammatory response. So it really does help to join up some really, really informative answers.
So are you saying that anyone in the UK could provide a fecal sample?
They could,
Mhm.
yes, and it would be helpful. And if you imagine that those kind of answers aligned to their questionnaire answers on Elaros, we'd be learning stuff. We'd be learning good stuff.
Okay, thank you. So what have you and your team learned so far about the practicalities of running a study of this scale across ME/CFS and Long COVID?
I think that hopefully we went into it, knowing the area and knowing what was involved. But just that it is challenging. I've alluded to some of the challenges already. That you've got to make sure that you're defining your patient groups very, very clearly. Otherwise, you're going to run into problems with your conclusions and with your peer review. You've got to get all the nuances of this past your ethics committee, not because we're doing anything bad or scary or intrusive or anything to people, but just because they're there to guard the lives and the sensitivities of the patients, and want to be seen that we do everything perfectly and by the book. And then we've got the challenge ahead of us that we're generating some very large, very complex data sets. And one of the things that reviewers always say to us is, but this will generate so much data and it's going to be so complex and so many different areas. You're talking about, Epstein-Barr viruses and other herpes viruses, and you're talking about antibodies, and you're talking about bacterial species, and you're talking about, all these different analyses. How on earth are you going to put them all together? So we've had to put already quite a lot of kind of brain wattage into that, and how we satisfy that criticism, and getting really clever people on board who are really good at big data analysis and integration of big data sets, so that it can really add value and be more than the sum of its parts, and lead to some useful answers. So so if you think, I guess what I'm trying to say is that we're trying to kind of balance all of those sort of big science demands with the real life demands of trying to remember that at the other end of this, there are some real life people who are struggling with their diagnosis and with this condition, and we're trying to be inclusive with them. We're trying to communicate with them well and just be fair to them and do something useful.
Thank you. It is exciting how much data you can get out of
Yeah.
this study, though. I think it's going to be fascinating to see. So what aspects of the study are you focusing on next?
So I guess what happens to us next is that we kind of count off the days when we can kind of move out of kind of business admin HR mode and go back to being real life lab scientists who are reopening the doors of a clinic, for example, to get some of these people through the door to to give their blood samples, to receive their jiffy bags through the post, to send them back to us, so that we can start doing what we love, which is doing analyses in the lab and beginning to get some answers. When I think about this field in general, and all the stuff that's going on in our lab, nationally, internationally, one of the phrases that I most use, or some people say most overuse, is tipping point. And I do think we are, as a field, at a kind of tipping point where all of these kind of vague mysteries are, I hope, heading to some kind of consensus of what on earth is going on, and what the underlying mechanisms are, and what the tests are that we could produce, and what the therapeutics are that we could try. And we're all just so desperate to get to the next stage and make some stuff happen.
Thank you. It does sound very exciting. I'm very much looking forward to what happens next and seeing some outputs. from the study. I know we're a long way off at the moment. I also really like to thank you for your time and telling us what's been going on so far in the Rosetta Stone study.
Great pleasure.
Thank you.